tract of ruminant like cow. They have a special alimentary canal. So, see, for example, when you want to lose weight, you want your weight to come down, mostly you eat vegetable foods, because you know vegetables and fruits, which they are some kinds of plants, they are full of cellulose. And cellulose in your body without any production of energy removing from the body by feces. So you use cellulose-rich food for losing weight, because you know you don't have the enzyme cellulase. But ruminants like cow, by eating plant, get weight for many hundred kilograms. The body of this animal and human, no difference, because both of them cannot produce cellulase. Both of them, human intestine and cow intestine, cannot change cellulose to acceptable substrates for intestine. But the main difference between ruminants like cow and human is the normal flora present in their intestines. Our normal flora look like our intestinal cells cannot break cellulose to glucose. But there are some members of microbes, bacteria, and protozoa present in the rumen of ruminants, which they have the ability of degradation of cellulose and changing cellulose to glucose, and glucose can be used for energy production. So human lose weight by eating food containing cellulose, but ruminant in the opposite direction not only not losing weight but also get weight and will be fat by eating plants. So now we understand that valuable normal flora are present in the rumen of ruminants. Climax community of the rumen are cellulose degrading bacteria. So there are some bacteria present there in sufficient numbers, which they can break cellulose by production of cellulase and change it to glucose, like Bacteroides and Raminococcus. But plants are not only cellulose. There are different kinds of compounds are present in plants which are eaten by these animals. Some of them like starch, human and cow, no difference. We don't have problem with starch. For example, when we eat potato as a plant, So we get weight because we don't have problem for breaking potato to substrates using for energy production. We don't have problem such as cows. But by the way, in ruminants like cows, as well as cellulose degrading bacteria, starch degrading bacteria are present like Selenomonas. And plant cells also contain some proteins. So whatever food come to the intestine of cow, respected bacteria are present, like protein degrading bacteria such as Veillonella. And also there are some methanogenic bacteria such as Methanobacterium. Not only valuable bacteria are there, but also some protozoa effective on cellulose are present. Cellulose and protein degrading protozoa like polyplastron. These items are some examples of those bacteria and protozoa which are effective on cellulose, protein, and the other things. So the other microbes and the other populations in other valuability are present also. So here we can see food is coming by eating by ruminant to the rumen. Then bacteria break it and change it to the useful substrates. Then bacteria are eaten by predators, protozoa. So there we can see food chain also. There is food chain. Food come to the ruminant, then transfer to bacteria, then transfer to protozoa. And it is interesting, at the end of intestine of ruminants, they can digest protozoa and receive those foods from this food chain. So ruminant cannot use cellulose. Bacteria use it and give substrate to ruminant. Then remaining food in bacteria transfer to protozoa by predation. Then predators are digested by ruminant and again receive those proteins, those things from protozoa. It is what? Why a small cow changing to big cow with many 100 kilograms? Because with high absorbance, different kinds of food which are eaten changing to energy for ruminant. So we see that predatory-prey relationship between bacterial population and protozoan populations are present. Protozoan population predominantly ciliates, apurate in the succession following development of complex bacterial community. Pioneer bacterial community modifies environment which with production of various volatile acids and removal oxygen, so allowing succession of prostate climax community. So the types of microbes present in the rumen of ruminant, not allowing another populations, another microbes present in many other animals to be present there. Now, after talking about different environment as intestine, which when they are newborn, it is virgin, and then pioneers microorganisms come, then first succession, second succession, and going to climax community. Now we want to talk about a definition related to the homeostasis. Homeostasis. What is that? See, suppose if some children at home like whatever mother prepare in the breakfast, in the lunch, in the dinner, if they like it, if they adapt themselves to what are prepared at home, so in parallel way, they are under protection of parents, because those children obey the rules of home. So when there is a habitat, like an ocean or soil or body, so during the month we eat something. So those microbes, like what you eat, they can be protected by our intestine. And those microbes which their phenotypes, genotypes, and metabolism and their enzymes are not matched with the food which we eat, they are not protected by our intestinal habitat. So homeostasis is the protection of environment for those microbes which are adapted to that environment. What is homeostasis? Homeostasis is the protection of environment related to those microbes adapted to whatever present on that habitat. Homeostasis protect those microbes like the temperature, like the pH, and likes the food which are present there. If they are like all the items of the environment protect them. If they don't like one or some or all of these items, environment don't protect them. So it is homeostasis. It is homeostasis. Sometimes maybe some changes shortly happening in the environment. Sometimes maybe some fluctuations occur in an environment. Suppose some toxins come to soil, or some petrol come to the sea. So it changes the homeostasis for a short time. Suppose one father every day take children to school, take them back, buy food, and do many things. If father for short time have problem, for example, have COVID-19 and go to hospital, homeostasis of home is changing for short time because father is not there. So for short time, there are many problems for going, come back, school, food, and security, all the things going to the problem. But immediately in the return of the father, all the things come to the normal situation like before. So if any changes happening to an environment, like for example, a ship containing petrol has accident in an ocean, an area going to problem till the time those petrols are in the water. When those petrols are cleaned and removed, because this homeostasis is the previous homeostasis, again protect the previous microorganism, the previous animals, and the previous living things. Homeostasis never change. Maybe there are some fluctuations, but after removing the fluctuations, all the things come back like the original time. So microbes adapt themselves to whatever present in the environment. Suppose in example number 1, if there are some microbes with ability to production and accumulation of substrate in an environment, like accumulation of nitrate or hydrogen sulfide in ecosystem, temporarily increase the population that can use this metabolic intermediates. But by the stop of production of this substrate, again this fluctuation terminate, and homeostasis is previous homeostasis. For a short time, those microbes can use this substrate are dominant, but by the end of this production, homeostasis is previous homeostasis, and previous microbes are present there again. Example 2, sometimes we have population shift. Sometimes we have population shift. Population shift mostly happening in response of environmental conditions such as seasonal light, temperature change. For example, suppose a river in the winter it is 5 degrees centigrade, and in the summer it is, for example, 30, 35. So changing the temperature during the year in one habitat is forcing changing the microbes. So sometimes homeostasis temporarily changing from time to time. So homeostasis of winter is different with homeostasis in the summer. And accordingly, we can see some changes, some succession, some changing the population from time to time. And it is repeated regularly because every year winter is coming after autumn. Never it comes after summer or before summer. So all the years Winter is in a specific time, spring in a specific time. So some changing in the microbes present in the environment repeated and repeated every year and a specific time. So accordingly, we can see population shift. Population shift may also occur in response to environmental conditions, such as seasonal light and temperature changes. For example, in North Pole, 6 months is darkness and 6 months sun shining. So when for a long time there is no light, some microbes disappear, photosynthetic microbes disappear. When 6 months there are light, photosynthetic microbes are coming. So it is population shift. Population shift according to the season. In one season, some microbes are present. In another season, another one. In a river, in cold condition, psychrophiles, and in summer, mesophils are dominant. In North Pole, during the darkness, there is no photosynthetic microbes in the water, and during spring, there are full of photosynthetic microbes. A metabolic niche filled by mesophilic population during summer season may be occupied by a psychrophilic population during winter. Such seasonal shift in populations are known to occur on a regular basis. For example, Vibrio parahemolyticus exhibits an annual cycle in estuaries. Its occurrence during summer and spring, but disappear during the winter. When winter is not suitable time for life and division, they go down to the sediment and try to pass this severe condition and reaching to the suitable time again. Some members of this bacterial population survive in sediment during winter, allowing for continue the cycle. If they remaining on the top water, they are killed by coldness. So they go down to the warmer place in the sediment by minimum metabolism, waiting for new good season. So according to these examples, we understood that there are some temporal niche. There are some temporal niche. Some microbes in the winter, they are in the sediment, so it is a niche. But in the summer, they leave this niche and going to the upper side of the water. It is new niche. So these microbes have two niches. One niche is in the upper side of the water, and one niche is down in the sediment. According to the season, they go somewhere which can continue their lives. microorganisms may occupy a niche in a habitat at one particular time, but not at another. Example, various algal population in many aquatic habitats exhibit seasonal successions. Under various temperature and light regimes that occur during each season, different populations fill the niches of the ecosystem. Right now, we want to have another example and to see succession changing the population during the year according seasonal fluctuations. Some seasonal population fluctuations can be viewed as repetitive succession towards a stable community. Example is related to the nearshore region of Arctic Ocean in North Pole, which it is very cold and many months there is no light and many months it is continuously sun shining. 1. Each spring, what is the spring? So when darkness after a long time finish, by starting sun shining, spring is starting. A spring is starting. So by presence of light, there are so many carbon dioxide and there is water, so photosynthetic microbes appear. Photosynthetic algae, photosynthetic bacteria, all absorb light and they do photosynthesis. And they are under the ice on the water. Because during the darkness it was so cold and the upper part of water covered with ice. So number 1, each spring large blooms of algae like trees blooms, you can see in the spring the trees are full of flowers, flowering. So in similar way, the multiplication, high multiplication of algae we call as blooms. 1. Each spring large blooms of algae on the underside of the sea ice is started. Blooms occur at the same time each year. Why? Because all the years at this time sunshine can be seen and sun rises every year at the same time. After algal bloom, after receiving light, carbon dioxide, and photosynthesis, and after large blooms, so in the water is full of organic foods. So what is photosynthesis? Photosynthesis is production of carbohydrates by use of carbon dioxide and sunlight in the presence of water and minerals. So now water is full of organic matters. So bacteria are coming now again, especially Flavobacterium and Microcyclus. As the ice melts, so the upper side of the sea ice as melts. Now it is the time for attack the predators and parasites and bacteria to the algae, and they want to use the whole algae as their foods. As the ice melts, the habitat is removed, ending community succession. Algal populations released into the water column diminish and consumed by predators or subjected to bacterial decomposition. Then light period is finished and winter is starting with a long time darkness. During winter, pigmented bacterial populations, photosynthetic bacteria look like algae disappear from the surface waters, but some algae and pigmented bacterial population survive in the sediment. So they go to another temporal niche, which it is in the base of the water, in the sediment. 5. When winter ice form again, it establishes a suitable habitat for recurrence of the seasonal succession. With return of sufficient sunlight again in new spring, algae colonize under the ice and process repeat itself again, repeating and repeating and repeating. So this succession every year by this manner is repeating because homeostasis is forced this way. Homeostasis, when there is light and the habitat is warmer, there is a homeostasis in favor of photosynthetic microbes, and when there is a large number of foods present by photosynthesis, it is in favor of heterotrophs. So some photosynthetic and heterotrophs come together to the water. And in the winter, homeostasis is not in the favor of photosynthesis. Sometimes severe or catastrophic environmental changes may overwhelm the ecosystem. Severe or catastrophic has the same meaning. Catastrophic means severe. Severe or catastrophic environmental changes may overwhelm the ecosystem's homeostatic controls, destroying existing community and initiating of a secondary succession process. A few minutes back, I had some example, and now again repeating. Suppose there is an island with one volcano. So volcanic fires come to the island, and these fires kill all animals, many microbes, and destroy many things. So for some time there is fluctuation in homeostasis. But when volcano is stopped, again homeostasis return back the previous microbes. Suppose you wash your body by soap and water, so it kills and removes many normal flora of the skin. But when you come out of washing, again homeostasis is previous homeostasis, and those removed normal flora again come back to live there. Suppose agriculture people add some herbicides or fungicides for killing them. As they stop it, again homeostasis revert back. Those previous microorganisms there. So severe or catastrophic environmental changes may overwhelm the ecosystem's homeostatic controls, destroying existing community and initiating of as secondary successional processes. Catastrophic events such as introduction of pollutants into aquatic or terrestrial ecosystem, volcanic eruption, fungicide application to soils and plants and whatever. So till the time a catastrophic or severe environmental change is there, there are some succession, some new populations come there, but why finish the severe or catastrophic environmental change? Again, homeostasis is previous homeostasis, and previous microbes again come back to that environment. They come back to that environment. After disturbance, homeostasis acts to restore the disrupted community, and once disrupting factor is removed, usually there is a secondary succession back to the original community. Washing the skin disrupts the microbial community of the habitat. Washing doesn't result in a new climate community, but rather initiates a succession back to the original microbial community. Similarly, the microbial community in agricultural soil returns to its original composition following tillage. Homeostasis also acts to restore the initial community when alien or alloctonous microorganisms enter habitat. So when there is a habitat, there are some microbes present there under protection of homeostasis. So those microbes which they are under the protection of homeostasis, they fight with non-native microbes according their space and their foods. So homeostasis also acts to restore the initial community when alien or alloctonous microorganisms enter the habitat. So every day we never eat sterile food. Sometimes some pathogens come with food to our intestine, but we don't feel them because our normal flora, according their space and according their foods, compete and try to remove them and never we feel them. Problem is coming when the number of pathogens are so much high. So when you prepare food at home, when you do cooking, so the number of pathogens are the minimum. When after eating food, the extra food you put in the refrigerator, it is for what? Why do you use refrigerator? Because you don't like the number of pathogens enhanced till the time of you eat the food. When the number of pathogens are very low, so normal flora easily, by protection of homeostasis, remove the pathogen, and we don't go through the problem. So homeostasis also acts to restore the initial community when alien or lictinous microorganisms enter that habitat. Many microorganisms enter gastrointestinal tract on food particles are quickly eliminated. And even after ingestion of microorganisms that cause disease, the climax microbial community eventually returns to normal. Similarly, lictinous microorganisms that enter soil and aquatic habitat are transient members of the microbial community, and antagonistic interactions act to remove the alien microbial population, thereby restoring the original composition of the community. Here the main question is that, before finish this title, if some populations are so much strong in the soil or in the water or in the intestine, why cannot exclude all the other populations? Why wherever we see powerful and weak populations are living together, why powerful microbes never can remove, eliminate, or exclude the weak populations? Or the question in better way, which factors preclude competitive exclusion and permit coexistence of the population? Number one is seasonal and diurnal fluctuations. So, for example, suppose two types of microbes are present in a river or in a sea. One is mesophil, the other is psychrophil. So they have different temporal niche. They are in the same habitat. Both of them are present in the ocean or in the sea, but not in the summer, not in the winter, not in any time of the year. Both of them are not in the same place. So in the winter, psychrophil come up and mesophil go down. In the summer, mesophyll come up and psychrophyll go down. So seasonal and diurnal fluctuation, the varying inputs, is one factor. The other factor, even though one population is stronger, but there are many different foods are available for different microbes in most habitats. So each find their own foods. There is no need, for example, one weak population to go through competition with one strong population. Each find its suitable food, and there are many different foods there. So it is number 2, multiplicity of resources and foods. Spatial heterogeneity of habitat. See, maybe in the intestine, one population is powerful, but the structure of the intestine, one part is stomach, one part is small intestine, one part is the large intestine, and each part has several another niches. So sometimes some powerful are in one niche, in one place of the habitat, the other weak one is in another part of the niche. For example, in the ocean, photosynthetic microbes on the top never going through the problems with those bacteria which are present on that habitat in the down or in the sediment. They are up and they are down. So there is heterogeneity of habitat. The other one is regulations by predators. Regulations by predators. When one group is powerful, so it cannot be in large numbers to remove all the other populations, because each population, even though they are strong or even though they are weak, they are under the control of predators and parasites. If one population is powerful, their numbers are controlled by effect of predators and parasites. And the last factor is dormancy. Sometimes one population is very powerful and the other population is under the pressure. So when the pressure is high, the weak population change their appearance from vegetative form to resistant form of spore or cyst. When the numbers of a strong group finished, again they return back to the vegetative form. So seasonal and diurnal fluctuations, multiplicity of resources, spatial heterogeneity of habitats, regulations by predators, dormancy are different items which is responsible. Many weak populations can live with many powerful populations together. And the weak populations are not removed from environment by effect of those powerful ones. The other title we are going through it is microbial interactions with animals. Microbial interactions with animals. And subtitle is microbial contribution to animal nutrition. Microbial contribution to animal nutrition. For human, till this part, we understood our normal flora is working as an army. And if any pathogens come, they attack the pathogens. Not allowing pathogens makes us going to the disease. If we are be careful, we cooking food well, we put food in the refrigerator before using, and if we be careful and those pathogens in very low number come to our intestine, our normal flora are sufficient to suppress them, to kill the pathogens. But little we hear about the effect of them on our nutrition. The most valuable example regarding to the role of microbes on the nutrition is related to herbivores like ruminants such as cow. Digestion within ruminants. So there are some ruminants like deer, mouse, antelope, giraffe, caribou, cow, sheep, goats. These ruminants have rumen. And rumen, as I explained a few minutes back, are full of those cellulose digesters. They can digest cellulose because they have cellulase. So the foods which come to the intestine, coming to the rumen of ruminants, is full of cellulose. Even human cannot use cellulose, animal also cannot use it. But if they receive energy from plants with high contents of cellulose, it is because of the cellulose, it is because of the cellulase production by microbes present in the rumen. Their foods are rich of cellulose, but they don't produce cellulase enzyme themselves. They are dependent on associated microbial population for degrading cellulose. Rumen contains large populations of protozoa and bacteria, contributing food digestion. So what happened? In human, when we eat plants, yes, we absorb antioxidant, we absorb some good substances, but we cannot use cellulose. And all cellulose go out from our body by feces. That in ruminant, those bacteria which have cellulase break the cellulose to glucose. And in the Roman, situation is anaerobic. 30 to 40 degrees centigrade and pH 5.5 to 7. So the glucose which is released by the help of enzyme cellulase is the form of glucose is going under the fermentation. If glucose is in our food, in human food, glucose is absorbed by intestine and goes to the cells. That in ruminants, after breaking cellulose, glucose is released, then glucose in the rumen fermented to several acids. Which types of acids? Acetic acid, propionic acid, butyric acid, as well as carbon dioxide, methane, and water. So in human, glucose goes to cell by help of blood and going through Krebs cycle and electron transport chain. But here we don't have glucose. Glucose is changing to the different low molecular acids such as acetic acid, propionic acid, and butyric acid. So cellulose, starch, and the others changing to carbon dioxide, water, methane, and low molecular acids such as acetic acid, propionic acid, and butyric acid. Organic acids are absorbed into bloodstream. So whatever acids are produced in the rumen are absorbed by blood vessels and they are transferred to different cells of the ruminant. Then in animal cells, they go through oxidative metabolism and aerobically it goes to the electron transport chain and all NADH2 and FADH2 are changing to the ATP. Another item is produced is methane. It is methane produced by methanogenic bacteria within rumen. But methane is not used by the animal and in the form of gas by mouse and accompanying with feces going out of the animal. So methane is useless. Production of methane is useless for the animal. Valuable protein, so plants contain high quantity of cellulose, but even though it has protein, lipid, and the other things. Protein, volatile protein is digested and absorbed in the lower portion of gastrointestinal tract rather than fermented to the methane. Anaerobic environment in rumen is present. Small percentage of caloric value of food, about 10%, is lost to animal during microbial digestion process. So 90% of the energy transfer from food to animal. It is very good. It is what we can see a small cow changing to many 100-kilogram cow after a few months. So what are we doing now? We are talking about microbial contribution to animal nutrition. So because of the presence of those microbes containing cellulase, high quantity of plants which are eaten by ruminants can be used as energy source for production of ATP. Rumen harbor a great diversity of microorganisms, like what? Like cellulose digesters, starch digesters, hemicellulose digesters, sugar fermenters, fatty acid utilizers, methanogenic bacteria, proteolytic bacteria, as well as lipolytic bacteria. What are their names? Their names such as Bacteroides, Raminococcus, Succinomonas, Methanobacterium, Butyriovibrio, Selenomonas, Succinovibrio, Streptococcus, Eubacterium, as well as Lactobacillus. Lactobacillus. Okay. So if you accept, because today I have so many lectures and your class is in better position than previous class, we follow the remaining one, the remaining items, only a few page is remaining, and 100% tonight we can finish it. So 6, I think, which time should we come back? We should come back on I'm checking. So at 6 o'clock, again, we will come back to finish all the remaining items. Now, something around 20 minutes. Okay? So have a break, 20 minutes. 6 o'clock we come back, and after maximum 1 hour, we finish whatever remaining. Okay, you can leave the class and have a short break. Thank you very much. See you soon after 20 minutes.